So this is part 4 of Chapter 5's lecture on biomolecules and carbon. And we were talking about there are two groups of nucleotides, both the purines and the pyrimidines. And so again, make sure that you know which ones belong to which. So adenine, guanine being the purines, cytosine, thymine, uracil being the pyrimidines. So the nucleotides, which are the monomers of DNA, they bind between the DNA strands. So when so there are two types of bonds that occur in a DNA molecule. So as you remember, DNA is a double-stranded molecule. And in between those two strands, we first of all have hydrogen bonds. And those hydrogen bonds always occur between a purine and a pyrimidine. And so, again, are different classifications of the types of bases that are and actually the bases define the nucleotide as either a purine or a pyrimidine, are really important because as you look at the graphic here showing the hydrogen bonds between a purine and a pyrimidine, you'll see how having them match up in this way allows us to keep an even distance between our two strands of DNA. Because, for example, if we had like two purines that were bonded together here, and then maybe two pyrimidines here, and two pyrimidines here, and two purines here, we would end up having our strands become wavy. So for some reason, it's important that these strands are kept in this straight, symmetrical shape. And so this structure has to do with some sort of function that is yet to be determined, really, but it certainly makes sense to have symmetry and a straight line of a double-stranded DNA. So another thing to know is the bond between adenine and thymine is a 2-hydrogen bond, and between guanine and cytosine, it's a 3-hydrogen bond. So why are hydrogen bonds important between the two different strands? This again is going back to your regular freshman biology class, but those hydrogen bonds are the weak bonds in biology. And so by having this weak bonding between the two strands, it makes it easy to unzip the DNA for replication or for transcription and then rezip it for storage. So the two types of nucleic acids we have again are RNA and DNA. And another important structural difference is DNA is the only one that's double helixed. So the only one that actually has that hydrogen bonding occurring. RNA is a single helix. And again, this is pointing out that the monomer of a nucleic acid is a nucleotide. I want to point out here that nucleosides, you may just see that sometimes and think maybe it's a typo, but it actually is a structure. And the difference between a nucleoside and a nucleotide is that a nucleoside simply does not have the phosphate group attached to it. So again, remember that a monomer of a nucleic acid is nucleotide, and it has 3 parts. So you can think of the T in tide as standing for 3 parts. So it has the base, the sugar, and the phosphate group. Whereas the nucleoside, again, does not have the phosphate group. Another thing to note here is that the atoms in both the nitrogenous base and in the sugar are numbered. So you'll see here we have the numbering system of this purine, and then we have the numbering system here in this ribose sugar, actually, on this one, because it has the oxygen. And the reason that we actually add the primes on the sugar is to distinguish between the numbering systems in these two molecules. So to build our nucleic acid, we need to polymerize our nucleotides into a chain of or a series of nucleotides. So I'm going to show you this really quick video here talking about how that's going to occur.
DNA stands for DNA.
So these phosphodiester bonds hold our strand of DNA or RNA together. And it's linking again, very importantly, you need to remember that it is linking together our 5' and 3' ends of our nucleotides. Because if you remember, when we're actually studying replication or even looking at transcription of DNA, um, we need to remember the direction of 5' to 3' in terms of reading the DNA and making the new DNA molecule or making our RNA molecule. And so when we're talking about that 3', 5' direction, what we're talking about is the carbons of our sugar molecules in our nucleotides. So again, remember that the first carbon is the one immediately to the right of the oxygen in the ring structure. The second would be going clockwise around. This is the third, the fourth, and then the fifth is attached to the phosphate group. So this is how we, again, are making these phosphodiester bonds. And remember that when we're building and reading our DNA, and actually we'll look at this in the next slides. When we're, for example, replicating DNA, you need to remember that DNA is complementary. And what that means, again, is that we have the complementary base pairs matching up. So with the G, we always have a C. With T, we always have an A. And so that is an important part of this complementary structure. The other part of that structure that's important is that one strand is 3' to 5', and the complementary strand here is then 5' to 3'. So these go in opposite directions. And that's a really important consideration when we're talking about matching up complementary strands of DNA. And again, remember that the 3' and 5' are talking about the actual structure of the nucleotide. So going back here again, when we're talking about it, it's 3' to 5' here, or 5' to 3'. So basically what it's showing you is in what orientation we have these molecules, because they're either going to be this way or they're going to be upside down, the complementary strand. So it's important. So this is a picture again showing you how replication is occurring. And we're going to go into replication in depth when we study molecular biology and start looking at DNA very closely. But here I just want to point out, for example, if I have a 5' to 3' strand of DNA and I'm making a new copy, I'm making a polymerized set of nucleotides. Each of these letters represents a nucleotide that is getting laid down, and it's going to be the opposite. So if it's T, I'm going to build my next nucleotide as an A, and then this T will mean this one will be an A, or this G will have now a C. So it actually dictates how these polymers are set out, and that's done through RNA or DNA polymerase when that enzyme actually sets these um, reactions, dehydration reactions, actually in place and form those phosphodiester bonds. Remember, we can also be saying that if we're given this 3' to 5', we can also say, this A means that we're going to have a T that's complement. This A is going to have a T that's a complement. So remember to have these line up together if you're figuring out how a polymerized sequence of nucleotides is actually going to be put together. So when does a cell actually go through replication? And there are just a few times really. It's when it's reproducing and going through mitosis, so actually during a growth phase, and also through meiosis when gametes are produced. So that's when the DNA needs to be copied. So cells going through reproduction and mitosis, that's only going to occur during growth again or else for repairs. So it's actually not happening all that much unless it's a portion of the organism that needs a lot of replacement of new cells. A couple interesting side notes is that the ratio of the A to T and G to C affects the stability of a DNA molecule. And what that means is that having 2 hydrogen bonds versus 3 hydrogen bonds, actually, it makes sense that there's going to be different energy requirements to break those apart. So from a biotechnology standpoint, if you have more of the GC bonds, which are the 3 hydrogen bonds, you're going to need a higher temperature to separate the strands of DNA. So what that ends up meaning is that organisms that are functioning at a higher temperature normally have more guanine cytosine base pairs in their DNA. And another kind of interesting note is that parasites tend to have more AT bonding, which is just the 2 hydrogen bonds. And it's actually not known why that is. Maybe you can be the one to figure that out sometime in your future. Another interesting note is that ATP, which you know is a very important part of a cell's functioning, is a modified nucleotide. So it has the same 3-part structure with a sugar, a nitrogenous base. It has adenine, and it has, instead of just one phosphate group, it actually has these 3 phosphates. And so I just thought that would be good for you to know. And we'll look at this more closely when we look at cellular respiration and photosynthesis. Okay, so next we're going to look at proteins. So proteins are multi-purpose molecules. They're the most structurally and functionally diverse group of molecules. They're involved in almost everything an organism does, and they account for more than, I'm running out of time here, they account for more than 50% of the dry mass of most cells. They function in terms of structural support. They are involved in storage, transport of molecules, in cell communication, in movement